So in this video, I'll be talking about the structure of DNA. I will make a future video talking about RNA, and then I'll also make sure to compare DNA and RNA because I know a lot of people get confused about the differences between these two, but I assure you if you watch my videos, you will 100% understand it. So let's get on to DNA. So let's first start off with an individual. In this case, this is a football player of Arsenal, okay? You can imagine this as anybody. And we know individuals are made up of the smallest unit that you can't see with the naked eye. You need a microscope. And that smallest unit is a cell, okay? So if you were to have a microscope and look at this person, you would see a whole bunch of these, a whole bunch of cells. Within a cell, there is many organelles, right? As you can see here. One of them being the nucleus. Inside the nucleus, surrounded by this membrane, this nuclear membrane, is DNA. DNA. Each person has DNA. And what exactly is DNA? So your textbook will tell you that DNA is the genetic material of living organisms. But in simple terms, let me try and explain this to those who don't really understand. So DNA is like your instruction manual. So pretend you order something online and it gets delivered to your house, say like a gym set. Sometimes they come with instruction manuals, right? To teach you how to build the specific equipment. So each equipment will come with a different instruction manual because they are meant to be built differently, right? So humans work the same way. All organisms work the same way. Essentially, DNA is our instruction manual. Your body will read this DNA like you can read a book. And then after reading it, it will build you. It will make the specific person you are. You will have a slightly different instruction manual from your friends, from dogs, from cats, whatever. All these organisms have slightly different instruction manuals. And based on your instruction manual, you'll be built. You will be made the way you are. Okay? That's basically what your DNA is. And it is hidden inside your nucleus of your cell. Each cell has your DNA inside, okay, in the nucleus. Now, we know we have so many cells in our body, but how long do you think a single DNA in one of your single cells are? So if you took one of your single cells and took out the DNA and stretched it out, how long do you think it can go in one single cell? So the answer is actually it can go 2 meters long. That's pretty crazy long considering how small a cell is. Now, if you were to take all the DNA from each cell of your body and put them next to each other in a row, it would go twice the diameter of the solar system, which is crazy long. And why is our DNA so long? So our DNA is so long because we're pretty complicated. We are not simple creatures. Imagine someone gave you the instruction manual to build a human. You wouldn't just get a page, right? You would get tons and tons of pages because humans are not simple like a simple machine. Therefore, the DNA is extremely long. Now that's it for the introduction. Now let's move on to talking about the specific structure. Because right now, this is what I'm showing you is DNA. This is DNA, but let's go into more zoomed in. So right now we already zoomed into that part of the cell, but let's zoom in a bit more until we can see the specific details. Okay, right, let's do this. Let's start off to explain this using the English language. So we know the English language is made up of letters, right? 26, we have a whole alphabet, okay? A whole alphabet of letters. 26, I think, I'm not sure, but I think it's 26. And if we arrange these letters differently with different lengths, we can get different words, right? But what if we change these letters around, we can get a completely different word. For example, look, silent is a word. If you rearrange these letters, you get a completely different word. Look called listen, right? Now, just bear with me. I'm just trying to explain this, so don't think I'm going off track here. So now, if you put a whole bunch of words together, you'll get a book, right? A movie, a story. But say you rearrange the words of that book, that would make a completely different story, right? For example, The Lion King. Maybe if you rearrange the words in Harry Potter, put them in different orders and all that, you'll get a completely different story, okay? Now where I'm getting with this is that our DNA is the same. The DNA of a human is made up of letters as well. Wait a sec. We also have letters. Just like the alphabet has 26 letters, A, B, C, D, all the way to Z, your DNA, this very molecule right here, only has, it's like a book, remember? Except this book, your DNA only consists of 4 letters. And I'll show them to you just now. There is G, and for now I'm just showing the letters, but I'll show you the exact pronunciation just now. So there's G, there's C, there's A, and there's T. Okay? G, C, A, T. And now, just like a book, depending how these letters are arranged in your DNA, you will get a different organism. For example, say you and your friend have different eye colors. This could be simply because one of your letters are swapped. Maybe in your friend it's CGTA, and that gives you brown eyes, and maybe in yours it is CTA. Maybe you have slightly different arrangement of your DNA. Your DNA is slightly different. People have different genetics, right? What could happen if, say, your DNA is a certain way? One example I just showed you is if you arrange the DNA, different order, you could get a slightly different trait. But in other cases, you could get a mutation, a disease. Sometimes your DNA mutates. I don't know if you've heard of that. Sometimes there's a mutation, and this causes sometimes a disability. Sometimes by swapping 2 letters of your DNA alphabet, you get a disease. Like maybe you can't breathe properly anymore. Maybe you can't carry blood properly anymore. Maybe your blood cells have different shapes. So bear in mind that if your DNA's order is changed, you will get a different outcome. Just like by changing the words of a story, you get a different story, right? So the same thing in our DNA with the organisms. Hope you're understanding everything so far. Let's keep going. Now, remember the DNA molecule I showed you on the first page. It looks similar to this double helix shape, right? Look at this image. It's not that clear, but it's the best one I could find. So look at this image right here. How many colors can you see here? Only 4 colors, right? You see the green, the orange, the gray, and the purpley, and they repeat, they don't change. There's no more colors. And that's exactly because there are only 4 colors, only 4 letters in the DNA alphabet. Okay, that's precisely why. Now also, look here. This is an image of this very molecule, this very DNA structure, just being folded open. So instead of continuing to double helix, they did fold it open to show you how it looks like. So you can see there's A, T, G, C, A, T, A, T, C, G, G, C. So there's no more than 4 letters. Literally, that's the most there is. That's the entire alphabet of your DNA. But see, they're in a specific order. This one is GC, next one is CG, and this repeats up and up and up and up. And different organisms will have a different order as it goes on. But what else do you notice? In this image, you may notice that only A and T go together, and only G and C go together. Never ever does A go with C or G. It's always like this. So that's one of the very things you have to remember. So I'll demonstrate this right down here. So again, here we have by the way, so remember how in the English alphabet we call these letters we call these things letters, right? English alphabet has a certain amount of letters. Well, in DNA, we call these not letters, but bases. So that's the way of saying letters in DNA. So your DNA consists of 4 bases repeating in different orders, okay? And like I just said, A and T always go together. G and C always go together, okay? Right now, up here, you may notice that there are 2 lines between A and T and 3 lines between C and G always. So don't worry about that. I'll explain that just now. We'll get to that. So just bear this in mind. I will explain why that is in a little bit. Ignore this line because that's going to be involved in explaining that. Now, what I promised you, so here are the exact names. They're easier to remember because they all end in "ine." You see what I mean? I-N-E. They all end in "ine." So there's guanine, guanine. There's adenine, guanine, adenine, and then thymine and cytosine. Lord. Okay? Try and remember these. They're not that hard because they all end the same, so but you need to know them. Okay. Good. Next. How can you remember, wait, let's get on to this. How can we remember that A and T always goes together and G and C always goes together? Because remember I said this is the only way that they can combine in your DNA. Well, there's an easy way to remember it. I remember it as an A and T always together. A and T, A for always, T for together. A and T are always together. Because if you know A and T are always together, then the remaining two must be together. So just by remembering A and T are always together, you will remember it, okay? So bear this in mind. This helped me remember it. Okay, let's get to this. So in the diagram that I showed you up here, you can see so far we covered all the color things, all the different bases, all the letters of this DNA. But we are still missing out this S kind of pentagon and this P kind of circle thing here that is part of this DNA, right? You can see they stay the same for every single row, right? You can say that every row that you get to, they're the same. It's just the letters that are changing. Just the DNA bases are changing. So let's cover what these things are. So by the way, first, and then the P, the S, and the A combined has a specific name. And this name is, it looks like this. So this is the letter, the P and the S combined you call a nucleotide. Let me get to this. Sorry. A nucleotide is basically the smallest unit of DNA. It's the thing that makes up DNA. So many of these combined will make up your entire DNA. Okay? So that's called the nucleotide. Just like, for example, when we eat carbs, carbs are all made of sugars. Just like carbohydrates, the big thing, is made up of many, many sugars. DNA is made up of many, many nucleotides. It's the smallest unit. Okay? You need to know what this is. There's a name for specifically this part of a nucleotide, the P and the S combined. I'll explain what these are just now, so don't worry. So the name for the P and the S is the backbone. And why do you think it's called the backbone? So when you think of your backbone, you can, in your body, you can think it's the thing that's holding your body together. Without your backbone, you just be a blob, right? You just like fall to the floor. So that's the same thing in your DNA. This backbone, this P and this S combination, is what holds these letters, is what holds the book together. Without this, your book would fall apart and the letters would just be floating everywhere, right? So this backbone, this P and S, is going to hold your letters together so that your body can be made, okay? It's making your structure stable. So it's making this very structure stable. It's like the backbone of it, okay? We're almost done here, almost done. So now to the very last part. So by the way, the smallest units, so this nucleotide, can also be called a monomer. A monomer, mono meaning one. So you can think it's the one thing that keeps, that is this one single unit that makes up DNA if it's repeated. So a monomer. So these monomers, how do they combine with each other to make DNA? Because remember I said this is the smallest unit and it combines with other ones to form your DNA, right? Like I showed in the picture above. So how exactly does it do that? So let's get to that. So let's first cover inside the molecule. You can see there are two bonds here between the sugar and the phosphate. The P is for phosphate, and this is the pentose sugar. Pentose because it has 5 edges. Pent means 5. So it's a pentose sugar, phosphate, and then your base. So what holds these together, these little bonds here, are covalent bonds. So these bonds are covalent bonds, and they're very, very strong. Okay? They're very, very strong. And the reason being is because they're supposed to hold your important molecules together. Okay? Now we're going to get to a bit of a trickier part, but if you focus, we can get this. So there's one more other bond that needs to happen. I'll put it here. I'll put the covalent bond here. One other bond, hydrogen bond, and you'll see why I made that part black. So okay, let's do this. Okay, almost there. So remember in the diagram above that I showed, two of these next to each other, you can see this one is upside down, right? It's running because it's going the opposite way. All of these are running down and all of these are going up. There's a specific name for that, which I'll get to. So in DNA, you got to know the one half runs downwards and the other half runs upwards, but they bond in the middle through the bases, the letters of your DNA. Okay? And how do they bond? So this A and this T is going to bond because remember, A and T are together. How are they going to bond? They're going to make two bonds, remember? Two bonds. Two what bonds? Two hydrogen bonds, okay? So hydrogen bonds is what happens between nucleotides, so between these two units. And covalent bonds is what happens inside the units for now. Okay, an easy way to remember that these are going to be hydrogen bonds because I made them dotted. The easy way to remember is that hydrogen bonds hide. H-Y-D, they hide. So as in they're not so prevalent. They're not so strong. They're kind of there, but they're not there. They're hiding. They're hydrogen bonds. So they're a bit weaker. So covalent bonds are much stronger, holding the entire molecule together. And these hydrogen bonds are kind of in between molecules. They're not that strong. Because remember, I told you mutations can happen. So these letters can swap and move around a little bit if sometimes there are mutations happen. And that's because this bond isn't as strong as this one. Okay, I'll put it here for you. Hydrogen bond. Easy to remember because it's hiding, it's not so strong, and it's in between these spaces. And by the way, I told you A and T have 2 hydrogen bonds, and G and C have 3 hydrogen bonds. Okay, so that's where the hydrogen bonds come in. And the reason why is because I'm not going to mention the details, but if you study chemistry, you'll know that adenine is a molecule that has 2 binding spots, and thymine also has 2 binding spots. And so they conveniently make 2 hydrogen bonds because they have those 2 binding spots. G and C, so guanine and cytosine, both have 3 binding spots on their molecule. So they conveniently also bind with each other. But A and G, or T and C, or whatever, they would not want to bind with each other because one would always be unhappy. Maybe A only needs 2 bonds, so it can make 2 bonds with guanine, but then guanine has 3 spots, so it has 1 missing spot, so it won't be so happy. That's why A and T always go together, just because they also share a common bonding number, and G and C also share their common bonding number. Okay? There's one part we're missing now, almost there. So remember, we have these 2 next to each other, but then we also have one under, and it keeps going on, right? It keeps going on. So how do these 2 that are below each other bind to each other? So an easy way to remember how they're going to bind to each other, because a lot of people find this tricky, is that consider each of these molecules, P, pentose sugar, adenine, they all need to make at least 2 bonds. Okay, so right now, this adenine has 2 bonds. I mean, it has 2 bonds, like it has this bond and this bond. So it's bonding to 2 separate things. It's already happy. This sugar has 2 bonds. It can have at least 2 bonds. And this P has 1 bond. So we know the P is going to need another one. So let's look at this one. This one, it will be happy when it binds to this one. The pentose is already happy, it's got 2, but the P only has 1 bond. So we want to make sure that the P has at least 2 bonds. So it's going to bond there. And now pretend we have another one below this. What will happen? So all of them are happy. Just pretend that these are binding to a molecule here, okay? So right now everything is happy. This A has its bonding with this one. It's bonding with this one. This one has 2 bonds, but this one still only has 1 bond. So we need to give it another one. So it has 2. So you can see the sugar in the middle, sometimes like it has 3, 1, 2, and 3. But the key is to remember, see, if we were to keep going, this phosphate would bind to another one and would keep going. So they will always have at least 2. So an easy way to remember that, just ignore those for now, is that each component, by component I mean A, the sugar, and P, needs to have binds to at least 2 components, 2 different components, okay? Because if you follow that rule and you accidentally do this, for example, maybe in the test you're like, oh, I forgot how this binds, and then maybe you do this, you think, oh, it's going to bind like this, then you remember, oh no, the rule I remembered was that they each need to bind at least 2, and at this moment, this one and this one has 2 bonds, this one only has 1, so it can't be right, this one needs to have 2 bonds, so then you'll be like, oh yeah, I remember, now it has 2 bonds, so now it's fine. Okay, that's how you're going to remember how, because a lot of the time they ask you to draw these molecules. Okay, so I'm going to copy this. So right now I'm going to make this clear here for you about, so adenine and thymine, 2 bonds. Cytidine, how many does G and C have? 3 bonds, remember? 3 bonds. Always. 3 hydrogen bonds. Perfect. Now very last thing, these 3 words you gotta remember: antiparallel, deoxyribose, and complementary base pairing. Antiparallel, I already explained. Antiparallel is is the way to describe the fact that the 2 DNA strands, these are a strand by the way, this is one strand and this is one strand. Antiparallel is the word to describe the fact that one strand runs upwards and the other strand runs downwards. So you can say DNA has antiparallel strands. The other one was complementary base pairing. Complementary base pairing means 2 bases, because remember these are bases, it's the way of saying letters in DNA. So 2 bases are complementing each other, they're similar, they complement each other, and therefore they bind together. So complementary base pairing. They complement each other because they both have 2 binding spots, 2 hydrogen bonding spots. So complementary base pairing. This is when the bases bind to each other. Deoxyribose. So the real name for DNA is deoxyribonucleic acid, DNA. Deoxyribonucleic acid. And the deoxyribose comes from the fact that the sugar is deoxyribose. Deoxyribose sugar. Okay? And this is important to know because a lot of the time in the test they ask you to distinguish between DNA and RNA, and you'll see in my next video how RNA is a bit different in this sense. So I will make sure to make some question videos. I'll get some questions, especially multiple choice ones from the IB, and I'll make a video on that so you can try and follow along. And do those questions, because if you can do those, you should be fine. If you watch this video, everything on every multiple choice question in the IB regarding this topic should be quite easy. So my next video, I'll be covering RNA. Make sure to watch that. I make it extremely clear because a lot of people don't understand RNA very well. And after that, you should get it really well. So